Hey, good afternoon, everyone. It's my pleasure to introduce Nancy Zhang, a colleague in Penn and also a good friend for many years. You can see her title there. Well, let me just also take it aside to say, some of you know she's visited us on Friday when we were in the summer. So now we give her a different treatment. Okay. Today's topic is more on the multi-omic integration, and the setting is the single-cell spatial analysis seminar series. So we have audience here in Forum Hall and 20 or more on Zoom. Let me quickly introduce Nancy's academic background. She received her PhD in statistics in Stanford in 2005. After a brief postdoc in UC Berkeley, she returned to Stanford in 2006 to be assistant professor in statistics. We actually overlapped there and collaborated on several interesting topics that continued, including change point detection and cancer subclone detection. She's now the Ge Li and Ning Zhao professor in statistics and data science. Nancy went to Penn in 2012 to also take up the position as a vice dean for the PhD program in the Wharton Business School. That's where she is still serving that important function. Nancy has received many awards, including the Sloan Fellowship and the Medallion Lectureship by the Institute of Mathematical Statistics. Her work has penetrated in many contemporary topics. And now in recent years, we've seen more from her group, both for herself and her former trainees, on single-cell analysis, on multi-omic integration, and on spatial analysis. So please join me in welcoming Nancy.
Thank you very much, June, and thank you all for having me here. I really enjoy now coming in person and meeting you all. And I'm excited to share this work. This is the first time I've given this talk. It is research that is still sort of on the grail, and we're hoping to get a paper out in a few weeks. So really welcome any feedback you might have. So I'm talking about single-cell signal recovery in single-cell data integration. Now, I know that this is our audience who work on single-cell and spatial data. So by a show of hands, how many people in the room have done some sort of single-cell or spatial data integration? So everybody, right? So almost everybody. So I'm just asking you, when you do single-cell data integration, you take many data sets together and get a single matrix and you do a UMAP, have you ever wondered to yourself, what am I erasing from the data when I do single-cell data integration? So that is what this talk is about. Okay, so I'm going to just bring everybody to the same page by starting with a concrete example. Okay, so here and I'll go back to this example later to show you the effects of data integration and our signal recovery. So here is a study that was published last year on type 1 diabetes, where we have cohorts of individuals, some healthy, some with type 1 diabetes, some with positive for beta cell antibodies. And we isolated their pancreas cells and cultured them and then sequenced them. Okay, and then brushing a lot of the details under the rug for now, because pancreas has many cell types, the alpha cells, the beta cells, we know that the beta cells are killed off during type 1 diabetes, and we hope to see that in our single cell data set. Now, if you were to go through, say, Seurat or Harmony or Liger or any of the single cell pipelines, right? So first of all, when you just do a straightforward UMAP, right, of the data and color them by subject, you see that the different subjects are all separated on the UMAP, and that shows you that there's batch effects, right? And then if you were to color them by the cohort, like disease versus no disease, you see that, yes, the different cohorts are different, but how much of those differences are due to these unwanted variation between your subjects? Okay, so questions a typical single cell study would want to answer is, what cell types are enriched or depleted within certain cohorts? Are there disease-specific cell states, and which genes or pathways are varying between cohorts? So these are pretty standard questions that I'm sure all of you have faced in your studies. Okay, so batch effects are in every study, and there has been a compendium of methods that have been developed to try to integrate data and remove batch effects, right? So this is a very active field of research. Going back 3 years, there was a benchmark study that was out that I think still today I see it as one of the most comprehensive benchmark studies, where they did a bake-off of the 10 methods that were sort of the most popular at that time, and 14 methods across 10 data sets, and they came to the conclusion that, say, Harmony, Liger, and Seurat3 are the most recommended methods, and say Harmony is more computationally efficient. So you could probably start with that, right? So I just want to say that this field has already seen a lot of progress with some good methods, and so far, even at 2023, I think most people use one of these top methods on this field. And these papers have garnered thousands of citations. It's very of high impact methods, right? So let's take this type 1 diabetes data set and let's integrate it, okay? And then let's say we apply Harmony as recommended by that benchmarking paper. And then you see, colored by cohort, the cohorts are kind of all on top of each other. And if you didn't color by cohort, if you color by patients, the patients were all now mixed together, right? So now this looks more interpretable. You can go around and start annotating the cell types, and the cell types do fall into discrete clusters. But then, the question now is, how do we know that what we remove from what you see on the left to get to what you see on the right is really just unwanted variation, the batch effects, right? So in order to answer that question, we have to go dig into the details and see how does data integration really work. Okay, so let's look at Harmony, which is what we applied. I mean, this is a short talk, so I'm just going to go quickly through and try to just go through the intuitive basics of these methods. So what Harmony does is it first starts with the cells and soft assign cells to clusters, and they have this term in their optimization function that favors clusters from a mixture of different batches. Okay, so they want clusters to ride across batches. And then within each cluster, they think that these mega clusters represent cell types, but then within the clusters, there's batch effects. So they try to get cluster centroids for each data set. And then they move the data set specific centroids to the centroids of the cluster to remove the batch effect. Okay, and then they move cells based on self-cluster membership, and then they keep on going. They do iteration, and this way they sort of massage their cells into clusters that are shared between batches. Okay, so that's Harmony. Seurat does something different. Every method has their angle on this problem. Seurat sort of takes two data sets and find mutual nearest neighbors, which are cells in one data set whose nearest neighbor is that other cell in that other data set whose nearest neighbor is itself. So these mutual nearest neighbors, and these are what they call anchors, and it uses these anchors to try to align the data sets. Okay, and then Liger came from Joshua's lab here, and then Liger takes a bit more principled approach. So it does this matrix decomposition where the cells are decomposed into K factors. These factors describe the cell state multiplied by a sum of a data specific loading matrix and a shared loading matrix. So this shared loading matrix is what it's trying to recover when it is integrating the data. And exactly how much of the data is shared versus not shared is controlled by this lambda parameter in its optimization function. So the lambda parameter controls how much of the variation to group under the dataset specific VIs. And that lambda parameter is chosen to maximize the alignment between the data. So that's a really great idea, but how do you, basically the lambda parameter is chosen so that the data sets align, right? But what if they are really biologically different? Okay, so going back to our original question, how do we know what we are removing is really batch effect? We actually don't. Okay, so it is more of an, you have done this yourself. It's sort of like close your eyes and wish everything to work, and it's sort of an art, right? So as June and I were talking about earlier on today, so a lot of the single cell analysis pipeline is more art than science at this point. And it is sort of that because we didn't have the kinds of data sets before to make it into a science. And what I mean by this is if you look at this benchmarking study that I was talking about earlier on in 2020, they actually tried to ask the question, how much of the biological variation is removed by existing integration data sets? So in existing methods. So they looked at 10 data sets, from human dendritic cells to mouse cell atlas, et cetera, et cetera. And then they did things like this, where they had the raw data, and here on the top, the raw data is colored by batch. There's 2 batches, blue and red. So this is actually one of the data sets. I think it was the first one, the human dendritic data set. So there's 2 batches, blue and red. And then there's actually these different cell types, 4 different cell types. And then so some of these cell types, are shared between batch. And then they would have this green cell type that is specific to the blue batch, and then this red cell type that is specific to the red batch. So these are biological differences between the batches. And then they want to see whether the green and red are mixed together, which they shouldn't be, versus separated. So in this case, Seurat mixed them together, which is bad, and then Harmony separated them, which is good, right? But these are very sort of, strong signals, right? So nowadays, a lot of single cell studies are done on disease, on treatment, and you don't expect a wholly new cell type to arise out of disease. Sometimes it does, but oftentimes it is more subtle signals, like a cell substrate that is enriched as such. So one thing I don't like about this benchmark is that they are looking at signals that are only very strong and not subtler cell states. And also, all of these methods by design were inspired by atlas building context. And what do I mean by atlas building context? You're basically trying to say, for example, build a brain atlas, in which case you might get a few samples, maybe from a few individuals. You don't really care at this point about the population-wide variation. You're getting a few samples, and then, or you're getting one sample and assay it using different protocols, and then you want to align like that. So if you look at this column right here, which is a number of batches that is tested on these data sets, the maximum number of samples they looked at is 5. So, at most these methods are aligning 5 samples, right? At least that's what they are benchmarked on. But recently they have been applied to these population scale large cohort studies, right? So the question is, if you have a large cohort single cell RNA-seq study or a spatial study, or what can you do that is better than these methods that were designed for these atlas building contexts? Okay, so so that was the that's the state of the art, I think, right now. So going back to our example in the beginning, right? So we have this pancreas, this study of atlas, study of type 1 diabetes, right? In this study, we actually have 11 healthy individuals, 5 individuals with type 1 diabetes, and 8 individuals with beta cell autoantibodies. Okay, so the existing integration methods treat them all the same. They ignore this stratification, right? And the control samples are only used post-integration for comparison. Now, the question to ask is, can we make use of the fact that these control samples are control samples during the integration process? Let me go to another example. So I'm going to show you 3 examples, and we're going to go through these 3 examples in our benchmark and illustrations. So here is an experiment done by my collaborators' lab, where they wanted to look at the effects of radiation on intestine, small intestine. So if they took mice and then they irradiated them, some like the FR and SR, the 2 different types of radiation treatment, and C is control mice, so 3 different strata. And then they, after treatment with radiation at day 2, 3.5, 10, 20, post-radiation, they took samples and sequenced them, okay? And the caveat here is that they had 2 technician teams during this experiment. And then, there's Clara and Yanis and Leo. And then when they did a UMAP of their data, these 2 teams completely separate on the UMAP. Now, I think many of us have met this kind of scenario before. It's actually, no matter how hard you try, sometimes, you get this happening, right? So the question they ask is, well, you know what? Maybe we can salvage the data because we included control mice during each of, in most of these experiments. Can we use those controls to help us in the integration of the data? Okay, so we'll come back to this later. Here's another example where it's a longitudinal trial. Okay, so here we have subjects. So it turns out these are cancer subjects, and they go through. They get treatment. In this case, it's an immunotherapy treatment. And at time 0, we get a sample from their blood, and then after treatment at different time points, we get more samples. So here, in fact, we had 10 patients. We took 4 time points per patient. We took peripheral blood cells and sorted for CD8 T cells. They thought they won't get that many batch effects. It's not a solid tissue, so there's no cell dissociation step involved. So it's just blood cells. And then, but then when you color it by sample on a UMAP, you still see, even, within the CD8 T cells, you see a lot of sample-specific clusters, okay? So they still needed to do integration. And one thing we ask is, well, you had this sample at baseline, right? So that was what they used post-integration, say, compare against baseline. But can we use the samples at baseline to get a sense about what is lost during integration? Okay, so, this is what we call a pool of controls. So what we realized is that in modern single cell and even spatial studies with VCM and some of the cheaper technologies, not so cheap, but, Codex, you're oftentimes, an experiment that is designed well should have samples that you can treat as baseline or control to compare other samples against. Okay, so before we go into the model that we're going to use, I'm just going to take to a sort of a thought experiment. Okay, so here we had, remember, 10 samples at baseline. So these are subjects that have not gone through treatment. So we took their samples. And then what we're going to do is we're going to pretend that 2 of them are treatment and the rest are controls. And then for the controls, we're going to remove a terminal effect, a subpopulation of the CD8 T cells. Okay, so we're going to remove the terminal effector CD8 T cells. And these are, so the T cells go through the stimulation, they differentiate, and then terminal effector T cells are sort of the last group that they get to. And then now, so these terminal effector T cells, we can imagine them as treatment-specific cell types. The reason why we want to do this is because the previous benchmarkings we've seen, they only did enrichment and depletion studies of major cell types. And now we're in a situation where we're looking at a subtype of CD8 T cells. They're not sort of they're not discrete clusters. If they're sort of, these are the naive T cells right here. I didn't label them, but that's what they were. So the terminal cells are right here. They don't even separate that well on the UMAP. Okay, so the question is, can we integrate without losing that treatment-specific cell type. So if you were to do so we tried Harmony because a lot of people use that. And so Harmony integrated all the samples, the 10 samples together. You see they're pretty well mixed here. And if you were to color by cell type, so we had this is with a immunology group, John Weary and Andy Min's lab. They're T cell experts, so they had specific markers that they used to annotate these T cells. And then you see that the terminal effector T cells are mixed in. So I'm going to show you how we can recover the signal from the Harmony integration to get them to separate out again. Okay, so that's sort of the goal of this talk. So now with that sort of building that stage for what we're going to do, here's an outline. I'm going to describe to you a model and algorithm that we call cell ANOVA. Okay, and then I'm going to describe benchmarks and validations on those 3 data sets to answer these 3 questions. Did we remove all of the unwanted variation? Okay. Did we do so while introducing minimal distortions? Okay, because integration always distorts the data. And then did we recover any biologically relevant information from the data that was lost? Okay. And then finally, because our model actually explicitly gives you the estimate of the batch effect on a cell and per gene basis, we're going to just ask the question, what comprises a batch effect? Because nobody really, they just integrate, but they don't say, okay, what did we remove? What is batch effect in single cell studies? I don't find a lot of guidance on that in the literature. So what we're going to do is look at what is batch effect. Okay, so first let's look at the model and the algorithm. So the model, actually, we build on existing methods because our method actually requires you to choose your favorite integration method. And we're going to try to recover the lost signal from that method. Okay, so you need to choose an existing method. And furthermore, you have to choose your control pool samples, meaning whatever your study design is, you have to designate a set of samples between, and I'll tell you exactly how these samples are used. So you can later on, maybe we can come back to this and you can think about how you would choose these samples. Okay, so given this control pool, then the cell ANOVA model is a linear matrix decomposition model, where we assume that each of these matrices is a sample. So we think of a batch as a sample. Each sample is a batch of cells. So each sample's data matrix is decomposed into a cell state encoding, which tells you, the macro state of a cell, multiplied by a mean variation, because the number of cells differ across studies. So the number of rows of these matrices are different, but the number of columns we assume to be the same because they are measured on the same features. So you have a common mean variation, a batch variation that is a sample-specific score matrix multiplied by a shared loading matrix, and remaining variation that we hope to estimate, that is embodying the biological signal that we want to recover. So this TWT is what W transpose is what we want to recover from the existing methods. Okay, and the V is the batch effect. Okay, so given this setup, now the question is, how do we estimate C, M, V, and W, and B and T? How do we estimate these from the data? Okay, so here is the strategy. And remember, I said you have to choose your favorite integration method. So, we chose Harmony, but you can use Seurat, Liger, whatever favorite method you have. So what we're going to do first is to estimate the cell state encoding matrix C. And the way we do that is we integrate all samples using your favorite method, say Harmony, and get row stacked Harmony aligned matrices X. So you align them and then you have a big big matrix with each row being a cell across all the cells in your data set. And then you can perform SVD on X to get the left singular matrices. That's the C, the left singular vectors. That's your cell state encoding. Now, these integration methods are not all that bad, right? They do preserve some of the variation. So some of the cross-sample variation is preserved in C, but others are lost. So what we're going to do next is then we're going to go back to the original raw data matrix for each sample, okay, the X's, the original data matrices. And then we're going to regress them again onto this state encoding matrix C. Do a regression to get the regression. So this is a K by P matrix. So, and then we take the mean of these RIs to get M. That's our mean variation. And now it's time to compute the batch latent space. So what we do now is we go to only our control pool samples. And then we take these RIs, which is the regression of the raw on the C, and then we compute the centered RIs on the controls, row stack them, so stack them across the samples and compute the SVD to get the right singular vectors. And that's the basis of our batch linear space. So if our batch effects lie in a linear space, the V is the basis vectors of that linear space. Okay, so that's our V hat. Now we know V hat. So how do we get rid of the batch effect? It's very easy, because we can rotate it out. What that means is that we can multiply our centered data matrices, centered by C times M, by 1I minus V transpose. That basically projects this data onto the null space of the batch effect. Okay, and then you add it back to your CM. So now you have a data matrix in the original dimensions of the data, a cell by G matrix that is removed of batch effect. Now remember, if you were to just go by your original integration and do a UMAP or do a linear embedding, that would be CM. CM is what you got before. You have added this recovered signal onto the CM. Okay, so guys, this is actually very simple when you think about it. Okay, these batch effects are both cell state and sample specific. They're cell state specific because it's C times B times V. For each cell state, you get a specific batch effect. They're sample specific because for each sample, you have a sample specific score matrix here in the B's. Yet the batch basis is estimated across samples, which is why you can estimate them across the control samples and then rotate them out of the other samples. Okay, so this takes a few minutes to do. We can also explicitly estimate the treatment effect after we remove the batch effect. And we also thought about iterating. So what it means is that now that we have the batch effect removed, can we get a better estimate of the cell state encoding and then repeat this process, iterate back and forth? We realize that iterating actually doesn't help here, because what it does is just, there's two places you can put your biological signal. You can put it into the cell state encoding, or you can put it into the loadings of the treatment matrix. Okay, you can say that my cell type actually had a shift in gene expression, or you can say I have a new substate and have a different enrichment of that cell state. The two statements give you the same observed matrix. So you could think about biological variation differently in terms of variation in the cell state encoding or in the effect size. Okay, so iterating would just change that attribution of biological signal between the C and the W. But in the end, if you're just looking at the batch removed matrix, it doesn't really matter. Okay, so here now we can have an explicit definition of batch effect. what do we mean by batch effect, right? So we think about it as technical variation between samples, but sometimes we group other unwanted biological variations such as circadian effects that we don't control for into batch effect. So now I can tell you explicitly what we mean by batch effect, okay? It is any variation between the control pool samples, between these gray samples, that was removed by the best effort initial integration by your choice of integration method. So your integration is going to remove a lot of biological variation. You don't care about the biological variation that is removed among the control samples. You want to only recover those that are removed from the treatment samples, because I'm using the control samples to estimate my V, my batch basis, right? So I'm removing them, okay? That doesn't mean that there's no preserved biological variation between the control samples. That's not true, because even healthy individuals have differences in their cell type compositions. If those are maintained in the original integration in the C matrix, then they are preserved. Okay? So I hope that gives you sort of a rundown, intuitive rundown of our method and the sort of the philosophy we take for batch effect removal and data integration. Okay, so now we do a benchmark and validations. So the first question we want to ask in benchmarking is sort of one about type 1 error, about whether we are removing all of the unwanted variation. Now, we did do a lot of simulations and other types of sort of data subsampling to answer these questions. But what we wanted to establish is, if you have a data set and you're doing integration with your data set, how do you answer these questions on the data set you are analyzing? Okay, because it doesn't matter if you do benchmarking other data sets that different cell types that are biologically very different or maybe technically very different. So how do you, given the data set you have right now, answer these 3 questions? Okay. So let's first look at the first question. Did we remove all of the unwanted variation? Okay. So in your data set, I'm assuming you have a control pool. So K control samples that you're willing to treat as control samples. Okay. So what you can do is to do jackknifing. So that's a statistics term. So what it is, it's a holdout of one control sample and treating it as a pseudo treatment, saying, I'm not going to use this as part of my control pool. Then you can apply the method on this data set, using the other K-1 control samples as a control pool. And then you can see whether an integration like that will effectively mix your treatment, your pseudo treatment sample with your control pool. Because you said that was a control, right? So you think that there really isn't much that is different for that sample. So an effective integration method should mix that with the other controls, even if it were not part of the control pool, right? So so you want to have a situation where you sort of when we compute this, this is quite common in single cell data. It's called the local inverse symptoms index. So what it is assessing the effective number of batches in each cell's neighborhood. So if that cell is in a neighborhood that is very well batch mixed, it should have a high I-Lc. If the cell is in a neighborhood that is not very, that's basically its own batch, then it should have a low I-Lc. So you want to be in a scenario where there's a high I-Lc, where the batches are well mixed. In this case, because these are all control samples, and you don't want a scenario where there is a treatment, pseudo treatment specific region in your data. So let's do this with your data, and let's see whether we get good mixing of that held out control sample. So what is plotted here is we hold this type 1 diabetes study, remember, had 11 controls. So we're gonna hold each one out, compute the I-Lc, and see whether we mix the batches as well as the state-of-the-art methods. Because remember, we're starting with the state-of-the-art methods and then we are recovering signal. So we don't want to introduce batch effects, right? So we want to have as good mixing as them. And then you see that cell ANOVA is the red one at the right. So you see that for this data set, except for the first sample where every method seems to do badly, something's wrong with that sample, but every other sample seems to be doing on par with the existing method. And that's what we want, because remember, our goal is to get as good mixing as existing methods, while recovering more signal. And then here is an immunotherapy trial, and here is the mouse radiation experiment. Remember, high ILC is good. That means better well mixing, better batch mixing. And then you see that for this data set, we only had 5 control samples. The question is, with 5 control samples, are you seeing enough diversity of batch effects? Is it representative of everything that can go wrong in your data? And it seems to be okay, although I would say that for this data, maybe we want to get a few more control samples, right? It's a bit lower for this sample, right? Okay, but you can do that diagnostic on your study. Okay, so then the other question is, the second question is, did we keep the mixing good while introducing minimal distortions, right? So what we mean by that is that when you do integration, and this has been brought out in previous papers, right? You always see what you observe is sort of truth plus noise. You remove the noise, but you might remove the noise while bringing in artifacts, like integration artifacts. So what you want to do is remove the noise, these black patches, and get something that looks like your ground truth, right? So we assess this by, for each cell, we compute the correlation for that cell before and after integration. Okay, so of course, integration is going to change things, but you want to have good mixing while introducing the least distortion, so having the highest correlation. Okay, and then the other thing, this is called global distortion. Then we can also look at distortion at the gene level, right? So this is looking across all genes. At the gene level, what you want is to recover the true differential expression between genes. So the ideally is that, in the ideal truth, there are genes that are cell type markers, and there are other genes that are not cell type markers. And then what you observe are noisy versions of that truth, right? So these starred genes are the cell type markers. They look different before you have batch effects. And then let's say you might have a integration method that sort of removes some of the noise, but introduce artifacts that make non-markers markers. So that's not good, right? And then you want a method that can remove the noise, but keeping good type 1, good p-values, realistic p-values. So let's look at cell type distortion, global distortion for now. Back to our 3 data sets. Okay, so you see that, and this was something that we were amazed that just by adding back that signal, that rotating out the batch effect and adding back that signal, we really correct the distortion in the data. Okay, so you see that all of these integration methods distort the data. Okay, it's unavoidable. But if you do a linear rotation to rotate out the batch effect, you get correlation that's close to 1. It's really nice. Okay, so we feel that we do really well in terms of getting good global, getting the least global distortion. Now, what about gene level distortion? How would you check that? So it turns out you can check that in your own study. Okay, so we go back to this idea of holding out one of the control samples. Okay, so pre-correction, pre-integration, you can look between cell types in that control sample and then do differential expression to get the cell type markers within that control sample. Now, there's only one batch there because it's only one sample. So those are sort of batch-free p-values for marker genes. Now, you can apply your method and integrate. And after integrate, you can take those same cells from that same holdout sample and compute differential expression again between the same cells. Now, it's exactly the same cells before and after integration. Now, before integration, there was no batch effect among those cells because they are from the same batch. So you would think that you want these p-values to look like these p-values, right? Because batch effect correction should only change things between batches. Does it? Right? So compute pre versus post batch, within batch p-values, pre versus post correction. Okay, so let's take a look. Again, our 3 studies. Remember, you can do this in your study, in the exact data set you want to draw inferences on. And then you can look at the preservation of p-values. You see that the horizontal axis is before, the vertical axis is after, and we just took one cell type, alpha cells, for each data set, but it looks the same across different cell types. You see that the linear method is our method, okay, so the cell ANOVA. It is on the identity line, meaning that we preserve the validity of the p-values for differential expression. And Symphony, which is a later update on Harmony, the reason why we use Symphony, because Symphony also is sort of in a sort of a reference alignment type of, it doesn't see the other cells before aligning this treatment cell to the reference. And then, so that we feel is more fair because it doesn't look at the data before computing the p-values. And so, but then still, it actually inflates the significance. So in this way, you can't tell whether a small p-value is a small p-value after integration. Okay, so I'm gonna skip over. I mean, this is just looking at different data sets and more cell types, and you see that the trend is it's very consistent. Okay, we preserve the validity of post-integration differential p-values by doing that signal recovery step. Okay, all 3 data sets, the same story. Okay, so did we do so while removing minimal distortions? I believe we correct the distortion. And then did we recover the biologically relevant variation? Now, this is the hardest question to answer because between samples, we don't know what is biologically relevant, right? So the way that, the way we did this is we made use of the fact that our studies all include meaningful subgroups within the treatment samples. What we mean is that, so for example, for type 1 diabetes study, there's the healthy individuals and the disease, but between the disease, there's those that are really type 1 diabetes and those that are only autoantibody positive, right? So those are 2 disease subcohorts. Now, we want, if this is a good method, we want it to be in a case where we remove the batch effect, but keeping the difference between, keeping any difference that exists between the disease subcohorts, right? The conditions, right? So if you don't have if you're too aggressive, you would mix the batches while also removing the condition effect. Whereas if you are not aggressive enough in integration, you would not remove the batch effect, and then you have a problem. So we want to sort of hit that sweet spot, right? Now, this is the same as what people have been doing in terms of preserving cell type separation after integration, but here we're not doing that. We're preserving the separation between the samples, between the samples that should be separated. So let's look at just, for a concrete example. So let's take, say your study has these 2 groups, AAB, the autoantibody positive, and type 1 diabetes. There are 2 different disease cohorts. Now, what we're going to do is devise a strategy where we can look at the degree of signal recovery between these cohorts. Okay, so what we do is for each cell, say a cell from this type 1 diabetes cohort, we're going to identify its out of batch nearest neighbors. What that means is we're going to look at its neighboring cells and identify those cells that are not from that specific batch that cell is from, that specific sample that cell is from, okay? We're gonna look at its out of batch neighbors, and then we ask how many of these nearest neighbors are from the same treatment group. So how many of the type 1 diabetes cells out of batch neighbors are also from type 1 diabetes patients, okay? So if the signal is removed between type 1 diabetes and autoantibody positive, then you will have a mixture. Whereas if the cell is within a type 1 diabetes specific neighborhood, then you will have a homogeneity in that neighborhood. Now, the reason why why are we only using out of batch nearest neighbors? Because if you were ineffective in removing the batch effect, you would also have an enrichment of its own condition, right? So you want to take the out of batch nearest neighbors to control. Okay, so let's look at, so this is a hard plot to read. So let me tell you what it is. So we look at, there's 3 groups here, right? Type 1 diabetes, autoantibody positive, and control. So we take each one as a center cell. So if the center cell is autoantibody positive, we want its neighbors to be enriched for autoantibody positive, right? Is that true? So we look at autoantibody positive neighbors. We see that autoantibody positive wins out, and also type 1 diabetes. And then control is last. Okay? So that's a biological signal. Is that all we hope to, can we do better? Right? So type 1 diabetes is really different. You see, so some of this, well, if there is not enough difference, if there is no biological difference between these 2 cohorts, then maybe you won't see anything. So here we saw a little bit, okay? And then the control is really different, okay? We see here that the control is sort of the autoantibody positive is being in between the control and the type 1 diabetes, which is what we expect, okay? Now let's look at because remember, we sort of built our method on harmony because we ran harmony first and then we covered on top of harmony. If you look at harmony, this is what it looks like. Okay, so this is the signal that we recovered. Okay, so let's look at the immunotherapy data. Here, I didn't tell you that there's actually 2 different immunotherapy treatments, treatment 1 and treatment 2. So right now we don't have to talk about exactly what they are. Okay, but there's 2 different treatments. And we want the treatment 1 cells to be enriched in treatment 1 neighbors, which is true. And treatment 2 cells to be enriched with treatment 2 neighbors, which is true. Now, what did it look like before signal recovery using just the first step integration, the best effort integration? Okay, oops. Let me show you. That's right. This one right here, right? So this was a treatment effect that we recovered from harmony. And this is the cycle effect, because remember, this is a longitudinal trial with 4 time points, right? So there's 3 cycles beyond the baseline. And then you see that in every case, we see more separation in the right direction as compared to what we started with in the initial integration. So you could do this on your data. You could integrate the data using whatever you want, and then you can do the signal recovery to see what was lost. Okay. So finally, just one last example. Here is the mouse radiation data. And remember, this is a case where there's 2 technician teams, right? Clara, Yanis, and Leo. And then their data is completely separated on the UMAP. So we want to integrate them. This is a case of a very strong batch effect. Now, here's the dilemma here, okay? Because from Leo, there is only 1 day where they use controls, day 3.5. And we want to integrate day 10 as well. There's no controls for day 10. So we're going to use the day 3.5. And hopefully by comparing day 3.5 of Leo to Clara Yanis samples, we can get a sense of what is this technical difference between these 2 technician teams. And we hope to use that to sort of appropriately integrate the day 10 sample from Leo. Make sense? So we want to see whether after integration, the day 10 sample from Leo is going to be integrated with the day 10 sample from Clara and Yanis. That's what we hope, right? We don't want this to be integrated with day 3.5 or day 20 or day 2, right? But then we don't have a control for that day. Is it going to work? Now, this is the same thing that the nearest neighbor, the out of batch nearest neighbors of the day 10 Leo sample, and how many of that is in day 10 for Clara Yanis? Most of them are. And then only a small portion of them are in day 2 and day 3.5 and day 20. So that shows that even when you don't include controls for every single condition you do, if you have enough of them, they allow you to estimate a diverse enough batch basis to remove the batch from those conditions where you didn't have control. You don't need control for every single condition. Okay? So I'm not sure how much time I have. Okay, so I think I'll skip this one. We basically, sometimes it's very hard to say that the subtle effect we found is real, right? So we had a case where we found in the immunotherapy trial where we did flow cytometry to identify cell proliferation state signature that was recovered by our method, and that checks with flow cytometry. Okay, so so I want to spend a few minutes to talk about batch effect, what comprises the batch effect, because we all wonder, right? What is it that's messing our experiment up? So remember that in our model, we have explicit terms for batch effect, right? The batch effect is C times B times V transpose, okay? And the batch effect is estimated using only this control pool samples post-integration. Okay, so remember, we define the batch effect as any variation between control pool samples that is erased by a best effort integration. It is really a mouthful, but we needed to say that to be precise about what we are treating as batch effect. Okay, so first let's just plot out that term, C times B times V transpose, across the 3 data sets. Okay, so that's just taking that term alone and doing a UMAP. And then you see that indeed they separate by batch very well because it's a batch effect. But then you see that within specific batches, like this brown batch here, it is also separated by sample. Sorry, by cell type. Okay, so that is the sort of explicit evidence that these unwanted technical variations do differ between cell types. Each cell type gets affected differently by the gymnastics of a single cell experiment. Okay, and then it's true across the 3 data sets. Like here is the CD8 T cell data set. Okay, they separate by batch as well as by cell type. Okay, and then here again, this is the mouse radiation experiment, separates out by batch and by cell type. But what's even more interesting is that if you look at the genes that are heavily weighted by V, so which genes really get affected by batch? Okay, and then if we look at the pathways that these genes are enriched in, for example, for the immunotherapy trial, you see these pathways. Okay, let's look at across the 3 data sets. Okay, so immunotherapy, type 1 diabetes, mouse radiation. And what you see here is that they share the same pathways. Okay, MYC targets, oxidative phosphorylation, unfolded protein response. This is very interesting because these 3 data sets come from 3 different labs. They come from 3 different cell types, very different cell types, 3 different tissues. One is in mouse, the others 2 are in human. And then the pancreatic cells underwent a culture step. But even so, they have the same top pathways that are affected by batch. So this is interesting, right? So this sort of invites the question, what if you don't have controls for your data? You might have, only a small budget or something. Can you borrow controls from other people's data, right? So we haven't went and did a systematic study, but that's where we're going to look next. Okay, so summary. Cell ANOVA model algorithm is really easy to do. It adds 5 minutes to any integration method. Okay, you have to have control. That's where the work comes from. You have to have control pool samples to estimate the latent batch variation space, and that allows you to rotate your data out of the batch variation space. We did a bunch of validations. We asked, did we remove all of the unwanted variation? Yes, at least as well as the current methods do them. Did we do so while introducing minimal distortions? That's where we really see an effect. We have much better data coming out of the pipeline. And then did we recover biologically relevant variation? Yes, and now we can have a very concrete, explicit statement to this. We recover biological variation that lies outside the span of the span of the batch variation space. And what comprises batch effects? Batch effects are indeed cell type specific. However, they share a lot of the same pathways regardless of where they come from. Okay, so I want to thank my collaborators for this work. The Zhao Junzhang is this PhD student, a very capable PhD student who did a lot of the real technical work. And Dongming Ma is my collaborator who had a lot of great insights on this linear decomposition model. And thank you.
Question from the room, Matthew, you will say, we'll repeat it.
I was curious if in your Villanova method, if it relates to paired full or mixed effect model, and if you could have for more complicated batching structure, and put it into sort of the more
Yeah. Yeah. Yeah, so Matthew asked in this model, can we accommodate more complex experimental designs using hierarchical structures, like for example, multiple batches or multiple control sets or treatment sets? Yes. So what I showed you is the simplest version, but sometimes what you might have is a control pool that is really small, only 2 or 3 samples that you don't expect to preserve any variation between them that is outside the integration. And then you have another control set, but you don't want to treat them as one big control set. So that you can certainly, it's very easy to extend the model to that case. And then you can have multiple treatment sets as well. Yeah. Yeah. You mean this NU and the estimation of this model, right? So so Dingjie asked, in this model, we have these C, M, B, V, and TW terms, right? So how do we guarantee convergence of the units of this, or the uniqueness, right? So there's identifiability, right? So for identifiability, we require this V to be orthogonal, the W to be orthogonal, the M to be orthogonal, and the BI's summed across samples I should be 0, and the TI's summed across samples I to be 0. So that guarantees the identifiability of these terms. Yeah, great question. The question was, we so far only in a single modality case, right, where the features are the same across these data sets. Have we looked at the case where the data sets to be integrated have come from different modalities? So we're doing that trying that currently, so I don't have a good answer as to how well this would work, but we're trying this model for integrating single cell, ATAC and single cell RNA. The problem here is that, there you have basically the cross modality difference, right? So you have to, I mean, I'm not sure how you would think, you have to have like what will be your control pool analog in that case. So the control pool should be somewhere where you don't expect differences between the modalities, right? So that's that, it's hard to imagine such a scenario between modalities, right? So it's very hard to look at that. What we have been looking at is single nuclei and single cell, right? So there is sort of two different, completely different protocols. But you can imagine cases where you say, this it should be, you don't care about the biological differences between them.
Is there a minimum number of controls needed for the method to work? What's the
Yeah.
relationship between number of controls recommended?
Yeah, yeah. So that's a great question. So we always think, and you should ask that about every data set, right? Do you have enough controls for this method to work? If you only have, we had 3 controls, 2 controls, can you still do this and think that you have removed the batch effect? Well, remember we had this, the first question we did, we remove all of the unwanted variation. We had this this exercise that we did where we did this leave one out control experiment, right? So here you take your K controls, like say 3, and then ask with the other, you can ask, leave one out and ask, do the other 2 controls integrate with this third one? Probably not, and that's your answer. Right? The thing is, for your data set, you can check this. This is something you can do on your data set to see whether your controls is enough, right?